Phages were discovered to be antibacterial agents and were used in the former Soviet Republic of Georgia during the 1920s and 1930s for treating bacterial infections. They had widespread use, including treatment of soldiers in the Red Army. However, they were abandoned for general use in the West for several reasons. Antibiotics were discovered and marketed widely. They were easier to make, store, and to prescribe. Medical trials of phages were carried out, but a basic lack of understanding raised questions about the validity of these trials. Publication of research in the Soviet Union was mainly in the Russian or Georgian languages and for many years, was not followed internationally. The use of phages has continued since the end of the Cold War in Russia, Georgia and elsewhere in Central and Eastern Europe. The first regulated, randomized, double-blind clinical trial was reported in the Journal of Wound Care in June 2009. Which evaluated the safety and efficacy of a bacteriophage cocktail to treat infected venous ulcers of the leg in human patients. The FDA approved the study as a phase A clinical trial. The study's results demonstrated the safety of therapeutic application of bacteriophages, but did not show efficacy. The authors explain that the use of certain chemicals that are part of standard wound care may have interfered with bacteriophage viability. Shortly after that, another controlled clinical trial in Western Europe was reported in the journal Clinical Otolaryngology in August 2009. The study concludes that bacteriophage preparations were safe and effective for treatment of chronic ear infections in humans. Additionally, there have been numerous animal and other experimental clinical trials evaluating the efficacy of bacteriophages for various diseases, such as infected burns and wounds, and cystic fibrosis-associated lung infections, among others. On the other hand, phages of Iniviridae have been shown to complicate biofilms involved in pneumonia and cystic fibrosis and to shelter the bacteria from drugs meant to eradicate disease, thus promoting persistent infection. Meanwhile, bacteriophage researchers have been developing engineered viruses to overcome antibiotic resistance, and engineering the phage genes responsible for coding enzymes that degrade the biofilm matrix, phage structural proteins, and the enzymes responsible for lysis of the bacterial cell wall. There have been results showing that T4 phages that are small in size and short-tailed can be helpful in detecting E. coli in the human body. Therapeutic efficacy of a phage cocktail was evaluated in a mice model with nasal infection of multidrug-resistant Abelmania. Mice treated with the phage cocktail showed a 2.3-fold higher survival rate than those untreated in 7 days post-infection. In 2017, a patient with a pancreas compromised by MDR Abelmania was put on several antibiotics. Despite this, the patient's health continued to deteriorate during a 4-month period. Without effective antibiotics, the patient was subjected to phage therapy using a phage cocktail containing 9 different phages that had been demonstrated to be effective against MDR Abelmania. Once on this therapy, the patient's downward clinical trajectory reversed, and returned to health. Bacterial quickly learned that bacteriophages are found wherever bacteria thrive, in sewers, in rivers that catch waste runoff from pipes, and in the stools of convalescent patients. This includes rivers traditionally thought to have healing powers, including India's Ganges River.